Hi, today I am going to present a special topic on spatial transcriptomics and spatial genomics, and somehow a basic comparison with normal NGS data set. So here, this is basically presenting the differences between spatial genomics and normal genomics and their applications. So the spatial genomics, as it is clear from the name that we are talking about the spatial location of the genes and their mutations, while when we are talking about genomics, then we are not specifying in which tissue or in which cell this mutation is located or this gene is expressed. But we are just saying overall gene expression is this or overall mutation is this. So this is what we are going to learn as a comparative study. For example, first difference table to represent the difference between these two kinds of techniques. Spatial genomics is new technique with respect to the normal NGS approaches. Spatial genomics or techniques, these techniques preserve spatial information, allowing researchers to see where in a tissue specific genes or mutations are located. It combines, for example, the spatial transcriptomics combines RNA-seq with a spatial barcoding or imaging. It allows mapping of gene expression patterns to a specific location in a tissue section. And it is useful for studying tissue architecture, tumor microenvironments, or developmental biology. Spatial genomics, similar idea, but focuses on DNA rather than RNA. It can map mutations, copy number alterations, and or chromatin structure in a spatial context. It is still an emerging field with fewer standardized methods than spatial transcriptomics. Now, about RNA-seq or genomics, these are bulk or single-cell techniques that can analyze gene expression or DNA sequences without any spatial information. RNA-seq or RNA sequencing measures gene expression by sequencing total RNA transcripts. It can be done on bulk tissue or single cells, that is scRNA sequencing. It loses a spatial context. You know which genes are expressed, but not where in the tissue they are expressed. Genomics refers to sequencing and analyzing the entire genome or DNA. It includes techniques like whole genome sequencing or whole exome sequencing. It also lacks a spatial resolution. Now, the key differences could be summarized. For example, RNA or DNA in normal genomics or RNA-seq. Spatial information not available. Resolution, bulk or single cell. Applications mainly for gene expression and mutations. Techniques, RNA-seq, WGS, WES, ChIP-seq, and more. Spatial transcriptomics and genomics, RNA or DNA, it could be analyzed. Of course, spatial information is available, and that is why this technique has been developed. Tissue level or single cell sequencing could be processed. Tissue architecture, cell niches could be understood. And techniques are 10x, Vision, Marefish. Spatial transcriptomics or genomics, not. We refer with a spatial context. While RNA-seq or genomics, it does not refer to any spatial context. This is key point. Now, the right side image here is representing the spatial transcriptomics and genomics, this one. And the chip or barcode looks like this. So these are the barcode which could capture 200 cells and their genes. And that could be visualized as heat map, that is sample example. And this is overall what you could feel about spatial genomics or transcriptomics. Now, if you want to read, like, for example, this tissue section with grid or barcodes, which you can see here, shows a thin slice of tissue. It could be tumor tissues or it could be normal tissue on a slide with a spatial barcodes. These dots are representing the barcodes. For example, it is here. Heat map overlay represents gene expression levels mapped back to their original locations in the tissues. Now, it highlights that this method preserves the spatial arrangement of gene expression. Implication, you can see which cells or regions are expressing specific genes, which is crucial for understanding tissue structure, disease, microenvironments, or developmental processes. Now, about RNA-seq, bulk tissue icon, this is, represents a sample taken from tissue, often homogenized. Now, test tubes, these things, symbolizes the RNA or DNA extraction and sequencing processes. No spatial context or label exists here. And it indicates that while gene expression or genomic data is obtained, the location of each gene's activity within the tissue is lost. Implication, you get a list of genes and their expression levels, but you don't know where in the tissue these genes were active. Now, next. In terms of application, spatial genomics or transcriptomics could be applied for tissue architecture mapping to visualize how gene expression varies across tissue region. It is crucial for understanding organ development and function. It is highly effective and beneficial to understand tumor microenvironment. It is used to identify spatial relationships between cancer cells, immune cells, and stroma cells. And it also supports immunotherapy research and precision oncology. It could be applied for developmental biology, tracks spatial gene expression during embryogenesis or organogenesis. It reveals how cells differentiate and organize spatially. Neurological research application, it could map gene expression across brain regions. It helps to understand brain function, neurodegeneration, and psychiatric disorders. It could be implemented for infectious disease and inflammation understanding to show how pathogens or immune responses are distributed in tissues. It is useful in studying diseases like tuberculosis or COVID-19. Now, for RNA-seq or genomics applications, which is quite common and well-known, gene expression profiling could be done to identify which genes are active in a tissue or cell. And it could be used for cancer research, immunology, neuroscience. Differential gene expression profiling could be done here also to compare gene expression between different conditions, for example, healthy versus diseased tissues or normal versus cancer tissues. It helps to identify biomarkers or therapeutic targets. Single-cell RNA-seq resolves gene expression at the individual cell level. It reveals cellular heterogeneity in complex tissues. Mutational and variant detection could be done, whole genome sequencing or whole exome sequencing. could help to identify mutations, SNPs, structural variants, and could be used in personalized medicine and genetic disease diagnosis. It could also be applied for transcript isoform discovery and it detects alternative splicing and novel transcripts. Now, this is the same thing which I have presented. So now if you want to understand more things, a spatial, like a kind of case studies. So for example, here, tumor microenvironment in medulloblastoma study. A 2025 study compared spatial transcriptomics techniques on medulloblastoma cryosections. The impact, it revealed how different tumor regions harbor distinct cell types and gene expression patterns, improving understanding of tumor heterogeneity and guiding the therapy. Brain mapping in mouse models. A study like a spatial transcriptomics was used to map gene expression across entire mouse brain. Impact helped in identifying spatially distinct neuronal subtypes and their roles in behavior and disease. It has been applied for inflammation in Crohn's disease study. Researchers applied spatial transcriptomics to intestinal biopsies from Crohn's disease patients. The impact, it identified localized immune cell infiltration and cytokine expression, offering insights into disease mechanisms and potential drug targets. Now, RNA-seq, which is quite common, so there is nothing